the resting membrane potential. All by itself, not hugely important, right? Because it's just sort of a phenomena. If you put a voltmeter across the cell membrane, you measure a difference. Well, the way the body turns this into something useful is by changing the membrane potential when things happen, all right? Now, the key to that change of membrane potential are these gated channels, all right? So if sodium channels can open and close, well, what makes them open and close? What happens when they open and close? So because neither sodium or potassium are at their nurse potential, both ions are sort of ready to move when we upset the equilibrium, when we open a channel or close a channel. When we open a potassium or sodium channel in the membrane, that molecule is going to move very quickly either into the cell or out of the cell down its concentration gradient. All right, so what are the triggers for these gates that open these channels? Well, the one we're going to talk about most is voltage. Okay, so we've been talking about the membrane voltage, the difference between the inside and outside. Well, electrical forces are big contributors to protein shape, right? So as the membrane potential changes, the shape of proteins embedded in the membrane also changes. So voltage all by itself can open or close channels. And that's the key to understanding the action potential that we're going to talk about in a minute. There are other causes, though. If all we had was voltage-gated channels, nothing would ever happen because there's nothing to make the voltage change, right? We need to have some other trigger to bring things about. So enter the chemical gate. So this is a classic receptor protein. Molecule, a ligand, binds to a receptor. That receptor causes, that binding causes a change in the shape of the protein and either opens or closes a channel. So one of the primary ways we see this happen in this system is with molecules called neurotransmitters, right? So you've all heard about neurotransmitters. Well, the way neurotransmitters affect neurons is by being chemically gated sodium or potassium channels, right? So serotonin, for example, binds to a receptor and causes a sodium channel to open, right? That sodium channel opening causes the membrane potential to change. That voltage change is then detected by other channels which open and propagate that change. So chemically gated ion channels are a key to neuronal and muscle physiology, right? And then the last kind are the mechanical channels. We depend on the mechanical channels to tell us about our external environment, right? So they're down here. So our sense of touch, for example, when something causes pressure on your skin, it deforms enough cell membranes that these mechanically gated channels are sort of pushed open. Sodium comes rushing in, that changes the membrane potential across this membrane, and that is detected by the cell and sent to the brain as pressure, right? Or touch, or a change in even internal pressure. So up here is an example of our chemically gated channels. So acetylcholine, for example, on the muscle cell membrane. Normally closed. When acetylcholine is present, this opens, allows sodium to rush in, changing the membrane potential. Once the membrane potential changes, well, now our voltage-gated channels can open or close accordingly. We're going to talk much more about this row when we get to action potential.